Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the law of genetics. So in this particular module, in the previous lecture, so far what we have discussed about the history or the development of the genetics field, and then we also discuss about the how the Gregor John Mendel performed the different types of experiments with the simple plant like the pea, and then what are the different characters what he has used, and based on these experiments he actually formulated the different types of laws which are been famously known as the law of inheritance. And in the previous lecture, we discussed about the first law, and we discussed about the law of dominance, and then we also discussed about the law of segregation as well. So, in the previous lecture, we discussed that when you are focusing only onto the one particular type of genotype and a particular type of phenotype which is related to that, it is actually going to govern the rule or the law of dominance, where one particular genotype or the trait is going to be dominant over the other traits, and that's why the trait which is going to be expressed in the first generation is going to be called as dominant traits, whereas the trait what is going to be expressed or not going to be shown as a phenotype is going to be called as recessive traits. Now, in the second generation, when these first generation individuals are going to be crossbreed, then there will be a segregation of the genotype, and as a result of that, it is actually going to show you the dominant traits and as well as for the recessive traits. Now, considering to this, we have also discussed about the exceptions of these laws, like where we have discussed about the incomplete dominance, we have discussed about the co-dominance and so on, and we have taken various examples which are actually the exception to this particular law, right? So where we have taken an examples of the blood groups, and we have also taken an examples of the mixing of the red and the white color and so on. Now, in today's lecture, we are actually going to discuss another interesting aspect where we are assuming that if the phenotype is going to be controlled by more than one gene, or if the you are going to study how the law is going to happen, or how the system is going to behave when you are actually going to deal with, with the two genes, right? Or the two particular type of genotypes. With single genotype, it is easy to understand because you are going to say, okay, the dominant trait is going to take over to the recessive traits. But when it is a two gene, then the system becomes more and more complicated, and that's how you are actually going to discover the new law, and that's how you are actually going to and that is what we are going to discuss in today's lecture. And in continuation to that, we are also going to discuss about how these what are the application of these law, and how these laws can be used to explain the different types of genetic variations what is being observed in the different types of organisms. Now, when you have the two genes, right? So remember that when we were talking about in the previous lecture, we said that it is actually a law which is inheritance for the single gene. Now we are going to move on with the more complicated system where we are saying that let's understand how the inheritance of the two genes or more than two genes is going to behave, right? Because you know that any type of phenotype is not being governed by a single gene in any organism, except very simple organism where you have the very small genotype, right? So when we talk about the inheritance of two genes, you are actually not going to behave with a single gene, but the cross products of these two genes. So let's understand about the two genes. So inheritance of the two genes is actually going to follow the different types of law, right? So Mendel has analyzed a number of crosses in which the two pair of alternative traits were involved. In each case, he obtained the same result, right? So from these experiments, he proposed his second law, that is the principle of independent assortment, which state that the factors for different pairs of traits assorted independently of one another. What this law means, that suppose you are dealing with two different types of traits. One is for the tall and the other one is for the color. So if you have the TT and YY, which is like YY is for the yellow color, right? Then the TT will not have any influence over the YY and YY will not have any influence over the TT, right? So and that's why the factor, because at the time of the Mendel's experiments, so he was not very sure about the genes and all these, genetic materials and so on. So he actually coined the term factor for defining these genotypes. So the factor for different pairs of traits assorted independently, which means when there will be a cell division, the TT is going to assort separately, the YY is going to be assort separately, right? There will be no influence of TT onto the YY or the YY onto the TT. So in modern terms, this means that the pair of alleles for one gene on different chromosomes segregate independently in the form of gametes. Consider an example, right? You have the seed shape and the surface, like surface could be smooth or the wrinkled and wrinkled, right? So the pair of traits would be seed color, yellow and the green, right? And you remember that the yellow is dominant over the green, and then when the Mendel made the cross between the two breeding smooth and the yellow plants, which means capital S and the capital Y to the wrinkled and the green plant, right? Remember that in the previous lecture we were talking about only with the one gene, that is the either the yellow plants or the smooth seeds and all that. But now we are taking about the two genes, right? So he got the result which is given, right, like this, right? So when you are doing a cross breeding between the original plant, that is the parent generation. So in the parent generation, you have the smooth yellow seeds or the wrinkled green seeds. So the genotype for these two would be like SSYY and the small s small yy, right? So in the genotypic level, you are actually going to have the SS, which is for the smooth, and the YY, which is for the yellow, right? And similarly, you are going to use the small ss and the small yy, which is for the wrinkled green seeds, right? Now, if you do the cross breeds, right, and you are going to produce the first generation, there's no problem. All the, you are going to produce s small s, capital Y, small y, which means in the F1 generation, it is actually going to follow the same law of dominance, right? And you are actually going to show the dominant traits. And what are the dominant traits? Dominant trait is the smooth and the yellow seeds. This means in the F1 generation, you are going to produce all the seeds which are going to be smooth and they are going to be yellow. Now, when the F1 is going to be crossed with the F1 cross breeding and you will produce the F2 generation, this means the smooth yellow seeds and smooth yellow seeds, but the genotype is different, like the small capital S, small s, capital Y, small y is going to breed with this, then you are actually going to produce the lot of combinations which are not going to be influenced by the other genotype. So what will happen into the F1 generation, right? You are actually going to produce the 4 different types of gametes. You are going to produce this 4 gametes and you are actually going to produce these 4 gametes. And when these 4 gametes will come together, right? For example, if 1 will come together, you are going to produce this particular genotype. When the 1 and 2 will come together, then you are going to produce this particular genotype. When the 1 and 3 is going to come, then you are going to produce. So this analysis is called as the checkerboard analysis, right? Where you are actually putting the genotype on one side and the genotype on the second side, and then you are putting a checkerboard, right? So if the SY is meeting with SY, then you are going to produce this. If the 1 and 1, this 1 and 2, this 1 and 3, this 1 and 4, this. Similarly, 2:1 is this, 2:2 is this, 2:3 is this, 2:4 is this. Because when you are doing the sexual reproduction, there's no reason that one will not breed with all of these 4, right? And similarly, the 3 is going to mix with 1, then you're going to have this. 3 is going to meet with 2, then you're going to have this. 3 with 3 is this, and 3 is 3 with. Similarly with the 4, remember that 4 is actually the small s small y, right? So small s small y, when it is matching with this, it is going to have this. 4 to 2 is this, 4 to 3 is this, and 4 to 4 is this, right? Now this is the genotype, this is the genotypic region, right? Now if you talk about the phenotypic, then out of remember this is the 16 offsprings, right, in the F2 generation. Out of the 16, 9 is going to be the smooth and the yellow seeds. Now this could be pure smooth and yellow seeds, or it could be a hybrid. For example, this is the pure one, right? This is the hybrid ones, right? So this is the pure for smooth one, but hybrid for the yellow, right? Similarly, this is the hybrid for the smooth one, pure for the yellow one, right? So even if the S is present singly or S is present doubly, it is actually going to show you a phenotype which is the smooth and yellow, right? Similarly, for this one also, right, see the S is capital S, but Y is Y, capital Y, small y. So whether the S is capital S is present alone, which means whether it is present like this or whether it present like this, it is actually going to give you the smooth seeds. Similarly, for the Y, whether it is present as capital Y or whether it is present as small y, it is actually going to show you the yellow seeds, right? So keeping these into consideration, out of the 16 seeds what you are going to produce into the F2 generation, the 9 is going to be yellow and the smooth seeds. Similarly, 3 out of 16 you are going to produce the smooth but the green seeds. So green you will produce only when it is going to show this particular type of phenotype, which means it is actually going to show you this particular type of phenotype. So smooth you will produce whether the S is present singly or in mixed with the small s, right? So smooth you will produce even here also, and smooth you are going to produce here also, and smooth you are going to produce here also, right? So for example, this one, right, this one is going to be a wrinkled yellow seeds, right? This is because the small s's coming together, this is corresponding to the wrinkled one. Similarly, you are going to have the other way around also, right? You are going to have, like, for example, this one, this is going to be wrinkled yellow, right? So keeping this into mind, right, 3 is going to be smooth green seeds. Similarly, 3 is going to be wrinkled yellow seeds, and 1 is going to be wrinkled green seeds, right? So that means out of the 16, you are going to have 9 which is smooth and yellow, 3 you are going to have smooth and green, 3 you are going to have wrinkled yellow, and 1 you are going to have a wrinkled green. So wrinkled green means you are going to have a small s and small y, okay? So that is this one, right? This is only going to be 1, right? So if I tell you about the genotypic ratios, then it is actually going to be 9 is to 3 is to 3 is to 1 into the as per the phenotypic ratio, right? And the genotypic ratio also, you are going to measure all this, and then you are going to tell you, right? So what it says that although we are talking about the two particular gene, one is for the smooth surfaces, one is for the color, but it is not going to influence the segregation, it is not going to influence the assortments. So all the F1 seeds from this cross were smooth and yellow as a result of the monohybrid cross as predicted, because this is going to rule the law of dominance. Then when these F1 generations are going to be, self-breed, then F1 generations are heterozygous for two pairs of alleles from the two different loci. Such individuals are called as dihybrid, and a cross between two of these dihybrid is called as dihybrid cross. So when the Mendel self-pollinated the dihybrid F1 plant to give the F2 generation, there were two possible outcomes. One, that the alleles determining the seed shape and seed color in the original parents would be transmitted together to the progeny. In this case, a phenotypic ratio of 3:1 is going to be predicted, right? So what it says that if there is a law of dominance and there will be no segregation, there will be no independent assortment, then the same phenotype will actually going to be, go into the next generation, and as a result of that, it is actually going to maintain the same ratio of phenotypic appearances, that is the 3:1, right? But that is not the case. So other possibility is that alleles determining the seed shape and seed color would be inherited independently of the one another. And in this case, the dihybrid F1 will produce all sort of genotypic and the phenotypic appearances, right? And as a result of that, you're going to have the 9 smooth yellow, 3 smooth green, 3 wrinkled green, and 1 as the wrinkled green. And this is going to be the pure. So this is the phenotypic ratio, this is the genotypic ratio, right? So that's why it is actually going to maintain a phenotypic ratio of 9:3:3:1, right? Now, according to the rule of probability, if the alleles for the two pairs of traits are inherited independently in a dihybrid cross, then the F2 from the F1 to F1 cross will give 9:3:1 ratio of the 4 possible phenotypic cases. Such a ratio is the result of the independent assortment of the two pairs of alleles for the two genes, right? And so 9:3:1 ratio may be considered as two separate 3:1 ratio multiplied together, the multiplication being done because of the product rule for the independent events. So if this is the 3:1 crossed by 3:1, that is actually going to give you the 9:3:1, right? So basically it is doing the same. It is actually working like a single gene behaving its own independently. So 3:1 is for the single gene, right? Remember that in the F2 generation, you are going to have the phenotypic ratio of 3:1. So since we are talking about the two genes, it is going to be 3:1 multiplied by 3:1, and that's how you are actually going to have the phenotypic ratio as 9:3:1. So further independent assortment in one example means that both pairs of traits involve the seed shape, seed colors are independent of one another, and in terms of the gene involved and how genes function is the generation of the phenotype. So this prediction was met in all the dihybrid Mendel cross, whatever he has used. Like, so he, remember that he has used the 7 different types of traits. So a combination also does not give any other ratio was as like 9:3:1, right? So in every dihybrid cross, it is going to give you this particular ratio, and this particular ratio is coming because 3:5, 3:1 is 3:1, right? So, for example, he counted 315 smooth yellow, 1058 smooth green, 101 wrinkled yellow, and 32 wrinkled green, and so very close to the predicted ratio. So Mendel, this result means that the factor determining the two pairs of traits can analyze the transmitted independently. Thus, in fact, the Mendel rejected the possibility that the factor for the two pairs of traits were inherited together. So this is all about the Mendel's law, right? Now, people will think that if this is all the case, then how the actually inheritance is working, right? So there are multiple theories which are actually explaining the inheritance phenomena, right? How the dominance is working against the recessive and so on. So one of the such theory is called as the chromosomal theory of inheritance. So what the chromosomal theory of inheritance is, right? So chromosomal theory of inheritance came very lately, because that time when the Mendel was doing all these experiments, and he was not very sure about what is chromosome, what is DNA and all that. Remember that he was using a term called factor, right? So factor means something which is carrying the information from one generation to the next generation, because even he was not aware about the gene also. So around the turn of 20th century, cytologists had established that within a given species, the total number of chromosomes is constant, right? Whereas the chromosome number varies widely among the species. For example, humans have 46 chromosomes, whereas cat has 72 chromosomes. So in the 1902, Walter and Theodore independently recognized that the transmission of chromosome from one generation to the next closely paralleled the pattern of inheritance of the Mendelian factor from one next generation to the next generation. This correlation has become known as the chromosomal theory of inheritance. So basically, what chromosomal theory is saying, that you are actually carrying the chromosomes, and these chromosomes are actually the genetic material, and that is going from one generation to the next generation, right? So the chromosomal theory state that the genes are located onto the chromosome and the two alleles of a genotypic segregation during anaphase 1 of the meiosis when the homologous chromosomes separate. The alleles may also segregate during the anaphase 2 of the meiosis if the crossing over take place, right? So this is what exactly it is saying, right? So you are actually going to have the heterozygous chromosome, right? So if two alleles are present, right, then it is going to be called as heterozygous. If the same type of alleles are present, then it is going to be called as homologous. So then in the S phase, it is going to be replicate, right? So then you are actually going to have the RR and RR small rr, right? And then you are actually going to have the three different possibilities, right? So if there will be no crossing over, right, then it will remain as like a pure strain, right? And within the anaphase, RR is going to be separated and it will form one gamete, and the small r is also going to be segregated, and then ultimately it is going to give you the in the after the anaphase 2, this is the meiosis, right? So you're going to have the four gametes, one which is for the capital R, the other one is for the capital R, then you are going to have the small r, right? So, and if there will be a crossing over, then there will be some portion of this and some portion of this, like some portion of R and some portion of small r are going to be mixed with each other, and that's what we have discussed when we were talking about the recombinations and crossing over and so on, right, in the previous lecture, right? So then it is actually going to give you a hybrid genotype, right? So R smaller, right? And when this is going to separate, it is actually going to give you the R small r, irrespective of that. This is here the capital R portion is less, here the small r portion is less, but and then in the anaphase 2 you are going to have the pure r, you are going to have the small r, where you are going to have some portion of the capital R. Similarly, you are going to have the a capital R, where you are going to have a small portion of the small r, and similarly you're going to have this, right? And then this is actually going to continue, and it will actually going to transmit the information from next generation to the next generation, right? So as the crossing over is actually bringing the more and more genetic variations, and whereas if you see that if there is no crossing over, it is actually going to remain as the the pure strains, right? So R is going to be remain as R instead of having the small r, right? And small r is also going to be remain as small r. It does not have any contamination of the capital R and so on. So crossing over is actually bringing additional diversity, right? Now the second question is how the sex determination works, right? How some organisms are male, some organisms are female. So that is always been done by the special class of chromosomes which is called as sex chromosome. So the chromosomal theory also explained how the sex is being determined in all different types of organisms. So the chromosome in eukaryote, which are not represented differentially in two sexes, are called as autosomes. So in the any organisms, you are actually having the two different types of chromosomes. One, the chromosome which are not different between the male and female, and these are called as autosomes. Whereas the chromosome which are actually going to be different, these are called as sex chromosomes. So whereas the sex chromosomes and the autosomes are found in all cells. So in a misconception of think that the sex chromosomes are found only into the gametes, but that is not true. Sex chromosomes are also being found into the somatic cells, but there you don't do any kind of development, right? Because the somatic cells only give you the tissue, it's not going to give you the any kind of gametes, and only gametes can actually participate into the reproductions, right? So sex chromosomes were discovered in 1900 by the Caroline McClung and Nettie Stevens and Wilson, right? And all experimenting with the insects independently obtained evidence that a particular chromosome determined the sex of an organism. And in the year of 1905, the Stevens found that in a grasshopper, the female has an even number of chromosomes while the male has an odd number of chromosomes. So there are two copies of the chromosome in the female but one copy in the male, and Stephen called that as extra chromosome is a X chromosome. Since this chromosome is directly related to the organism of sex of the organism, the X chromosome is an example of the sex chromosome. The sex of progeny grasshopper, then it is determined by whether a sperm contains X chromosome or it is not containing the X chromosome. So if it contains the X chromosome, it is going to be a female. If it does not contain the X chromosome, it is going to be male. So all eggs having a one X chromosome, if the sperm carries an X chromosome, then the resulting fertilized egg will have the pair of X chromosome and that will give rise to a female. If the sperm does not have an X chromosome, the fertilized egg will have the unpaired X and that will give rise to a male. Unlike grasshoppers, some insects have the two different types of sex chromosomes. For example, Stephen found that in common mealworms like the Tinobria molitor, the male has a pair partner chromosome for the X chromosomes. So that partner is much smaller than the clearly distinguishable the X chromosome, and Stephen called the partner chromosome as the Y chromosome, and like the X chromosome, it is a sex chromosome. So sperm cells of the mealworm contain either the X chromosome or a Y chromosome, and the sex of the offspring is determined by the type of sperm that fertilizes the X chromosome bearing the X. So if the double X XS mealworms are females and XY mealworms are male. So similar XY chromosome complements are found in other organisms including humans and a fruit fly Drosophila melanogaster. In some cases, the females have two X chromosomes and she is XX, and the respect to the X chromosome, and the male has a X chromosome and a Y chromosome. That's why it is been called as XY. So that is what it is going to show here, right? In the P generation, you have the parent 1 and you are going to have parent 2. And this is the example of the fly, right? So with female is actually going to have a genotype which is double X, whereas the male has a genotype which is XY. So remember that this is, we are only talking about the sex chromosome. There will be a other set of chromosomes, but that is going to be not different between the male and the female. Now in the when the progeny comes, right, so it is actually going to have the XX or the XY chromosome, right, because XX is going, when there will be a meiosis, it is going to form the two different types of gametes, right. This one also is going to form the two different types of gametes, right, and the F is, which is X is matching with X, it is going to produce this. If the X is matching with Y, then it is actually going to produce this, right. So the progeny genotype would be half XY, half XY, and that's why there will be a 50% probability that you are going to have the females, and there will be a 50% male, right? Because what genotype you are going to get? You are going to get XX, XY, XX, XY, right? That means 50% male, 50% female, right? So the pattern of the transmission of X and Y chromosome from generation to generation is very straightforward, right? In this figure, the X is represented by a straight structure, much like a forward slash mark, and the Y is represented by a similar structure by hook, right? So the female produce only X bearing gametes and the male produce both X and the Y. So random fusion of the male and female gamete produces the progeny with the XX and the XY splice. Now, there is another phenomena which is called as the sex linkage. So evidence to support the chromosomal theory of inheritance comes in 1910 when the Thomas Hunt Morgan of Columbia University reported the result of genetic experiment with the Drosophila, right? So Morgan received the Nobel Prize for his discovery concerning the role displayed by the chromosome in the heredity. So one of his true breeding strains, Morgan found a male fly that has a white eye instead of the brick red eye characteristic of the wild type. The term wild type refers to a strain and organism which is most prevalent into the wild type, right? So population of the organism with respect to the genotype and phenotype. For example, a Drosophila strain with all wild type alleles of the gene that determine the eye color has a bricked red eye, right? Variation of a white light strain also arises from the mutation theory, mutational changes of the wild type alleles that produce the mutant alleles that result in strain the mutant characteristics. So mutant alleles may be recessive or dominant to the wild type allele. For example, the mutant alleles that cause the white eye is recessive to the wild type alleles. So Morgan crossed the white eye male with a red eye female, and from the same strain and found that all the F1 flies were red eyes, right? He concluded that the white eye trait is recessive. Next, he allowed the F1 progeny to interbreed and counted 3,470 red-eyed and 782 white-eyed flies into the F1 generation. The number of individuals with the recessive phenotype was too small to fit the Mendelian 3:1 ratio. Later, Morgan determined that the lower than expected number of flies with the recessive phenotype was the result of the lower viability of the white eye flies. So in addition, Morgan also noted that the white eye flies were all males, right? So this was a novel result. This result in the all genetic cross performed with the other mutant type. Till date, the mutant phenotype had never been confined just to one sex. So this is what it is shown here, right? So in the P1 generation, the red color eye flies is being breed with the white color flies, and in the F1 generation, what you are going to produce, you are going to produce the XWY and XW, right? Whereas this one, right, and it is all going to be red eye because the red is dominant over the white, right? And as a result of this, the F1 genotype would be half, the XY, XX, and XY, and all these are going to be half female, right? So where W indicates the white eye alleles and W plus indicate the red eye, right? So it's going to be all red eyes, right? Similarly, when you take the F1 generation and when you do the dihybrid cross, or you are going to do the cross in the F1 generations, then it is actually going to have the XW plus or XW, right, versus the XW plus and Y, right. So this X is for the female, and so in this one, what you see here is that it is actually going to follow the phenotype of 3:1, right, because the three are red eyes and one is white eyes, right. So white eye is always been appearing with the Y chromosome, which means it is actually going to be a male, versus all these are females, right. So this is also female, this is also female, and this is also going to be male, right. So in the F2 genotype, you are actually going to have all these kind of genotypes, right. So you're going to have one as the WW, one as W plus WW, and this is going to be the W plus and Y, and this is going to be one W small y, right. So in the F1 phenotype, 3/4 are going to be red eye. So you're going to have two males and one female, whereas 1/4 white eye is going to be female, right. So this is all about the the X-linked characters, right. So then we also did the X-linked inheritance of the red eye and the white eye in the Drosophila melanogaster, right. And what he found is that, so Morgan actually, based on these experiments, Morgan proposed that the gene for the red eye variant is located onto the X chromosome. So the condition of X-linked gene is male is said to be hemizygous because the gene is present only once in the organism and there is no homologous gene onto the Y. So for example, the white eye Drosophila male have an X chromosome with a white allele, and though no other alleles that gene in their genotype. So these males are hemizygous for the white alleles. Because the white alleles of the gene is recessive, the original white eye male must have had the recessive alleles for the white eye on his X chromosome, right? So the red eye female came from a two breeding strain, so both of the X chromosome must have carried the dominant allele for the red eye, which is the W plus, right? The F1 flies are produced in the following way. The male receives their only X chromosome from other mother, and hence have the W plus allele and are red shapes. Whereas the F1 females receive a dominant W plus allele from their mother and a recessive W allele from their father, making them also red eyes, right. Then we have the F2, in the F2 produced by the interbreeding the F1 flies, the male that receives an X chromosome with a Y allele from their mother are white eyes. Those that receive the X chromosome with a W plus allele are red eyes. The gene transmission shown in the cross from a male parent to a female offspring to a male grandchild is called as the Crisscross inheritance. Morgan also crossed a true breeding white-eyed female, that is homozygous for the W allele, and a red-eyed male hemizygous for the W allele, and the cross is the reciprocal cross of the Morgan's first cross, that is the white male into the red female. All the F1 female receives a W plus bearing X from their father and a W plus bearing X from their mother. Consequently, they are heterozygous, that is the W plus W, and have a red eye because the red is the dominant trait versus the white, which is a recessive trait. All the F1 male receiving a W bearing X chromosome from mother and a Y from their father, so they have a white eye, right? So basically, this is the trait which is linked to the X chromosome, and that's why when if they are receiving a W which is bearing a X from the mother and Y which is from the father, right? Because this particular trait is not present on the Y chromosome, it is only present on the X chromosome, so it is actually going to show you the white eye. Furthermore, all the results obtained are different from the normal result of a reciprocal cross because of the inheritance pattern of the X chromosomes. So, inbreeding of the F1 flies involved a W capital Y male and a W plus W female, giving approximately equal number of male and female of the red and white eye flies into the F2 generation. This ratio different from the 3:1 ratio of red eye versus the white eye flies obtained into the first cross, in which none of the female and approximately half the male exhibited the white eye phenotype. The difference in the phenotypic ratio in the two set of cross reflects the transmission pattern of sex chromosome and the genes they contain. So Morgan's cross of Drosophila involved eye color characteristic, and we know now that the coded for by a gene is found only onto the X chromosome. So basically the red eye color the characteristics or the phenotype is being controlled by a gene which is only present onto the X chromosome. So if it is the X present on the X chromosome, then it is only going to express. So these characteristics and the gene that give rise them as a sex linked, or more correctly the X linked, because the gene locus is present only onto the X chromosome. So X-linked inheritance is the term used for the pattern of hereditary transmission of X-linked genes. When the result of reciprocal cross are not the same and the different ratios are seen from the two sexes of a offspring, the sex-linked trait may also be involved. By comparison, the result of the reciprocal cross are always the same when they involve the gene located on the autosomes with the same distribution of the dominant and recessive phenotype in males and the females. Most significantly, Morgan results that the inheritance pattern of a W gene paralleled the inheritance pattern of the X chromosome strongly support the hypothesis that the genes were located onto the chromosome. Morgan found many other examples of the gene on the X chromosome in Drosophila and in other organisms, thereby showing and his observations were not confined only to the single species, and it is actually a general phenomenon, right? So general phenomena is that if the genes are present onto the particular type of sex chromosome, whether it is a X chromosome or the Y chromosome, we have taken an example only at the X-linked characteristic, it is going to be transmitted as the X is going from one generation to the next generation. Now this transmission is always been linked to the linkage and as well as the recombination. So what is mean by the linkage? Genes on the non-homologous chromosomes assort independently during the meiosis, right? So in many instances, the certain genes are inherited together because they are located onto the same chromosome. So genes that are on the same chromosome are called to be synthetic, right? So genes that do not appear to be assort independently because they are located onto the same chromosome exhibit the linkage and are called as the linked gene, and these genes belong to the linkage group, right? So genetic analysis is the dissection of the structure and function of the genetic material. In classical genetic analysis, progeny from the crosses between the parents with the different genetic characteristics are analyzed to determine the frequency in which the different parent alleles are associated in a new combinations. The progeny showing the parental combination of alleles are called as parentals, and the progeny showing the non-parental are called as recombinants. So basically, the linkage is a phenomena where the two genes are their movement from one generation to the next generation are linked to each other because they are all present on the same chromosome. So remember that when there will be a transmission of a chromosome from the one generation to the next generation, it's not like you're going to get 50% chromosome, right, of a single chromosome, right? You will get a single 50% of the total chromosome, but the single chromosome, like the chromosome number 11, for example, you will get the total 11. It's not like you will get 11 by 2, right? So if the particular gene is present on the 11th chromosome, you will be actually going to, those two genes will move together, and that's how they are actually being called as the linked genes, right? So the process by which the recombinants are produced is called as the genetic recombination. This anyway we have discussed in detail, right? And through the test crosses, we can determine which genes are linked to each other and can then construct a linkage map or the genetic map of the each chromosome, right? So when you do these kind of test cross and the dominant cross and those kind of crosses, you will know that which genes are moving together, and that's how you can be able to make the particular type of map, okay, that these are the linked gene, these are going to show you the linkage. So classical genetic mapping has provided the information that is useful in many aspects of the genetic sample. For example, knowing the location of a gene on chromosome has been useful in recombinant DNA research in an experiment directing towards the understanding the DNA sequence in and around genes. Genetic maps are constructed using both gene markers and the DNA markers. A marker or the genetic marker is another name for a mutation or variation that gives the distinguishable phenotype. In other words, it is allele that makes a chromosome or a gene. Gene markers are alleles of the gene, whereas the DNA markers are the molecular marker, that is the DNA region in the gene that is polymorphic, and thus can be detected by the molecular analysis of a DNA. Genes on the same chromosomes are like passenger on a charter bus, right? So they travel together and ultimately arrive at the same destination. So this is a very, very important and classical example. If you and your friend is traveling on a bus, right, and if you are both sitting on the same, so wherever this bus is going, right, whether it is going to one organism or whether it is you will actually, you both will go and travel together, right, as long as the destination is same, right? So destination is same for both of these, right? However, gene occasionally switch from one homologous chromosome to another through the process of crossing over, right? So crossing over produces the recombination. It breaks up the association of the gene imposed by the linkage. So it happens that you are sitting on this bus, right, and the another person is sitting on another bus, and over the course of time, when actually it stops to one stop, right, then you basically go from this place to this place, and that is what is being called as the crossing over, right? So basically you can transfer yourself from this bus to this bus, and that's how you can actually be able to break the linked genes, right? One linked gene is sitting here and they were both together, right? But at this stoppage, there's a crossing over, right? So these two buses were going together, right? And so this is, imagine that this is the bus number 1, this is the bus number 2. So one of the gene which goes from this place to this place, and that's how now these two people are going to be in an independent route, right? So this particular bus is going on to somewhere else, this bus is going to somewhere else, right? So basically, once the crossing over happens and the linked genes are being separated from each other, then their movement from one generation to the next generation will not be together, they will be going to be separate. So crossing over refer to the exchange of gene between the non-sister chromatids. With the understanding of how the linkage affects heredity, we can analyze the crosses for linked gene and successfully predict the type of progeny that will be produced. For example, if you have the the two particular type of homologous chromosomes, one has the capital A and capital B, the other one is having the small a and small b. So this is going to be right. So you have one chromosome, you have another chromosome. So here you have the A, B, A, B. So this is a homologous two pairs of chromosomes. Similarly, you have the similar kind of another chromosome. So if there will be a cross breeding, right, then A will get the A from here and the B is going to get the B from here, right? And that's how you are actually going to generate the, so in the F1, this is what the progeny, the genotype, what you are going to generate. Now in the, if each allele pair, that is the A and small a, is always on a different homologous chromosome. So they must be written on the opposite side of the line. Thus a genotype like capital A or small a is incorrect, as the A and small a can not be on the same chromosome. Always keep the same gene order in both sides of the line. Writing something like AB by small a is correct, as it wrongly says that A and B are allelic at the same time. So this is all is shown here. So this is the F1 generation, the situation is. So you basically have to write like this, right? Right. So crossing over with the linked gene, right? Linkage is rarely complete. Usually there is some crossing over between the linked gene and producing a new combination of traits. Crossing over, which take place in the prophase 1 of the meiosis, is the exchange of genetic material between the non-sister chromatid, which is the non. After a single crossing over has take place, the two chromatids that did not participate in crossing over are unchanged. Gametes that receive these chromatids are non-recombinants. The other two chromatids which did participate in crossing over now contains the new combination of alleles. Gametes that receive these chromatids are called recombinants, right? So basically what happened is that you're going to have the, for example, if you are going with bus number 1, bus number 2, right? And if there will be an exchange of passengers between the two buses, right? Then you have two possibilities. One, this bus is remain as such, right, and there is no exchange of buses. But if there will be exchange of passengers between this and this, then you are actually going to have the another set of chromosomes where you are actually going to have some genes from this chromosome, some genes from this chromosome, and that's how after the crossing over you are so this is actually going to be the wild type, and this is actually going to be a recombinant strain. So this is going to be a new strain because it has the passenger from this side and it is also going to have the passenger on its own, right? So that's why it is actually going to show you the new phenotype, because this is going to have two different types of genes or gene portions, right? And so for in each meiosis a single crossing over take place, and then the two non-recombinant gametes and the two recombinant gamete will be produced. Remember that you are actually going to produce the 4 gametes in the meiosis, right? So 4 gametes can come together and can give you the 2 non-recombinant ones and the 2 recombinant ones, right? This result is the same as that produced by the independent assortment. So when the crossing over between the two loci placed in meiosis, it is impossible to determine whether the genes were linked and crossing over took place or whether the genes are on a different chromosome. So this is what is shown here. So if these are the two chromosomes, right? So this is actually the bus number 1, this is the bus number 2, right? And this bus actually contains the different types of genes. So if there will be no crossing over, it will remain as this, and it is actually going to produce the gametes like 1, 1 and 2, 2. This means it is going to produce the pure gametes of the 4 types. 1 for belonging to this blue one, the 2 which is belonging to this one, right? Similarly, if there will be a crossing over, then there will be an exchange of material between these two individual chromosomes, and as a result of this, it is actually going to produce the one which is pure one, 2 which is the pure one, but these two are actually participating into the crossover. So this is actually going to be 1, 2, and this is going to be 2, 1, right? Because this one is actually more portion of 1, very small portion of 2. Similarly, here you are going to have the more portion of 2 and the small. So this is actually going to be new this is going to be new species or new molecules, right, or new organism. This is going to be wild type organisms, and this is going to be new organism because the crossing over is being take place. So assume that we have genes that are linked and some crossing over take place between them. Suppose a genetic carried out the cross, right? So you are actually going to have these, right? So you are going to have the M versus small m and the capital D versus small d. And if you do a crossover, right? If the crossover take place, then the gene for the leaf type and the height, two of the four gametes produced will be recombinants. If there will be no crossing over, all four results gamete will be non-recombinant, which means if there will be no crossing over, then it is actually going to follow the classical mammalian genetics, and it is going to give you the pure non-recombinants. Thus, all over the majority of gametes will be non-recombinant. These gametes then unite with produced by the homozygous recessive parents, which only contain the recessive alleles, result into the mostly non-recombinant progeny and a few recombinant progeny. So this is what is shown here, right? So in this cross, you see that the 55 of the test cross progeny have normal leaves and all are tall, and 50 have a malted leaf and are dwarf. The plants are non-recombinant progeny containing the original combination of the traits that were present into the parents. So of the 123 progeny, right, that is what is shown here, right. So out of the 123 progenies, right, what is being shown here, right. So if there will be no crossing over, it is actually going to maintain the pure gametes, right, M and D and small m and small d. But if there will be a crossing over, then the MD and small d and M small d and the small m small d, it is actually going to produce the different types of variations, and as a result, it is actually going to produce the, these are the non-recombinant progeny where you are going to have the pure organisms. But when there will be a recombination, then it is actually going to produce the dwarf and as well as the tall. So this is going to be normal leaf, but it is going to be a dwarf, but it is going to be a malted leaf, but it's going to be tall because there will be a recombination. So this is what exactly shown here, right? So if there will be 123 progeny, 15th will have the new combination of that were not seen in the parents. 8 are the normal leaves and the dwarf, and 7 are the mottle leaf and a tall. So these plants are the recombinant progeny, right? So out of 123, the very small portion is going to show you the recombinations. So only 15 is going to show you the recombinations, where 8 is going to be the normal leaf and the dwarf, whereas 7 are actually mottle leaves and the tall. So a test cross for the two independent assorted gene is expected to produce a 1:1:1 phenotypic ratio into the progeny. The progeny of this cross clearly do not exhibit such a ratio, so we might suspect that the genes are not assorting independently. When the linked genes undergo crossover, the result is mostly non-recombinant progeny and fewer are recombinant progeny. The results is thus we observe among the progeny of the test cross illustrated, so that two genes show the evidence of linkage with the same chromosome. So this is the examples how you can be able to calculate the recombination frequency. So for calculating the recombinant frequency, what you are going to do is you are going to put the number of recombinant progeny divided by total number of progeny multiplied by 100, right? So this is simple, right? Whatever the number of progeny you got as the recombinant, you multiply that by, for example, 15, that is what we found in the previous examples, divided by 123 multiplied by 100, and that is what is shown here, right? So this is the 8, 1 and 7, right? So 15 is the recombinant one, and that's when you multiply, the 12% is the recombination frequency. So that's a 12% is the the recombination frequencies. So this is very simple and very easy, right? Then we also have the multiple genes inheritance, right? So how the multiple genes, right? So far we discussed about the single gene, we discussed about the two genes. Now imagine a situation where you are actually going to have the multiple genes, right? More than two genes, right? So how the inheritance of the more than two genes actually works. So in a polygenic inheritance is commonly known as quantitative inheritance and the multiple gene inheritance, where you are going to monitor multiple parameters, like for example, height, skin pigmentations, eye color, hair color, milk and egg production are the phenotypic characters which are present in the plants and animals. So many traits and phenotypic characters are inherited by the alleles present in the different loci, such as the polygenic inheritance. It is a type of inheritance controlled by more than one gene, where the dominant alleles have a cumulative effects and each dominant alleles express a part or the unit of the traits. For example, the skin color, right? The skin color is being controlled by the many type of genes, right? So every gene is producing one product, and the multiple genes, when they are coming together, the mixture of these products is actually going to give you what will be the color of the skin, right? The combined effect of these genes show a significant effect, but a single gene may not have little effect on the phenotype. Thus, root is called as the dominant alleles are present. The father of phenotypic inheritance is called as Coulter and the East we discovered as the polygenic inheritance. For example, the kernel color in wheat. So, how the multiple genes act on a characteristic can produce a continuous range of phenotype. Let us example first demonstration of the polygenic inheritance. So, Nelson actually studied the kernel color in the wheat and found that the intensity of the red pigmentation was determined by the three unlinked loci, each of which had the two alleles. Then the several homozygous variation of the wheat that differed in color. Like for example, Mendel, he performed the crosses between these homozygous variants and studied the ratio of the phenotype into the progeny. In one experiment, he crossed a variety of wheat that possesses the white kernel with a variety of possessed purple kernel and obtain the following results. So this is the parent generation, where you are going to have the plant with the white kernel or the plant with the purple kernel, right? Remember that. And then in the F1 generation, you are going to have the plant which is going to show you the red color kernel, right? And in the F2 generation, again, there will be assortment, right? So it is actually going to show you out of 16, right? It is going to show you one plant which is going to have a purple kernel, which is means wild type, right? This is going to show you the parents, right? Then 4 out of 16 is going to be dark red color kernels. Then 6 out of these is red color. Then 4 to 16 is light red color kernels, and 1 is to 16 is going to be white kernels. So this is actually going to be the pure breed, just like parents. This is also going to be pure, which is just like the parents. And these three are actually the different combinations where the different types of genes are present, right? Now, the scientists interpreted the phenotypic ratio as a result of segregation of alleles on the two loci. So he found that the alleles are the three loci that affected the kernel color, the two variations used in this process. So differently only on the two of the loci. So he proposed that there were two alleles at each loci, one that produced the red pigment and the one that produced the no pigment, right? So he will design that alleles that encode the pigment is going to be A positive and the B positive, and the allele that encode no pigment is called as A minus and B minus, right? So this is going to be a genotype where you're going to have the A positive and the B positive, and you're going to have a A minus and a B minus, right? The effect of these genes were additive, right? So each gene seems to contribute equally to color. So the overall phenotype could be determined by adding the effect of all the genotype as shown in table, right? So if there will be A plus A plus B plus B, then it is going to have the 4, right? That means it is going to give you the purple color kernel. If the A and B are actually coming together, then it is going to give you the pigmentation which is going to be dark red. So this is actually says that you are actually going to have the 4 of A and 4 of B. This means you are putting more of A phenotype. So it is actually going to give you dark red. Similarly, if you are adding more of this, right, A minus and B minus, then it is actually going to lower down the color, and that's how it is actually going to give you the red color. if you keep increasing the, because a minus is, a minus b minus is actually for the no color, a plus and b plus is actually for the red color, right? So if you, the combination actually is going to tell you which what color it is going to form. So if the a positive and b positive is less and a minus and b minus is more, then it is actually going to show you the light red color. If none of the A positive and B positive is present, only minus minus is present, then it is actually going to give you a white color or no color actually. So from these results, we see that the 5 phenotypes are possible when the alleles of the two influence the phenotype and the effect of genes are additive. When alleles at two loci influence the phenotype, more phenotypes are possible, and this would make the color appear to be very continuously between the white and purple. So there are multiple combinations which could be possible depending upon the amount of gene what is being present in a particular genotype, and that actually is going to decide the final color. The environmental factor had influenced the characteristics individual of the same genotype, and that's how it is actually going to affect, the appearances of the different types of color. And these environmental effects are always being studied in a separate field which is called as the epigenetics, where you are actually going to study how the environmental factor are actually going to regulate the appearances of the different types of phenotype, right? Then the environment played a little role in determining the kernel color, right? And a few loci encoded color. So Nelson was distinguished among the different types of phenotypic characters. This ability allowed them to see the Mendelian nature of the characteristics. So this is all about the Mendel's law of inheritance. What we have discussed so far, we have discussed about how the inheritance law actually governs the phenomena of the two genes inheritance and so on. And then we also discussed about the different types of sex-linked characters. We have also discussed about the how the X-linked character is moving from whenever there is a X chromosome present and so on. And we also discuss about the polygenic inheritance, and then we also discuss about the chromosomal theory of inheritance. So with this brief discussion about the Mendelian law of inheritance, I would like to conclude the lecture here. In our subsequent lecture, we are going to discuss some more aspects of the genetics. Thank you.